So thank you very much to the Society for inviting me to give this WDM Paton Historical Lecture. And the title, as Humphrey told you, is John Vane, Pharmacologist. The story begins, for us anyway, in 1946. A young chemistry graduate with his degree clutched in his hand was at a crossroads in his life. He had absolutely no incentive to remain in chemistry. He didn't enjoy the subject. He had little motivation, if any, to remain in science. And he didn't know what to do next. 40 years later, he was a highly fated pharmacologist. He'd won the Nobel Prize, the Lasker Prize. He'd been knighted for services to science. He'd published 500 papers, and he'd received 50 or more fellowships, medals, or high distinctions. So really, my talk is, what was it that happened in those years in between which caused such a radical shift in John's outlook and fortunes? So in order to do that, I thought we'd go back a bit to his childhood and school days. We often look back at these with 2020 retrospective vision and say, ah yes, well, we could have always known about his greatness in those days. What we do know is, and I guess this may strike a chord with many members of the Society, that his early interest in science was because he received from his parents a chemistry set. And he enjoyed playing with this, and after one spectacular disaster with a Bunsen burner in the kitchen, his father put up a shed for him in the garden, and between those four walls, he writes later, I became an enthusiastic and lifelong experimenter. And a little bit later, as he attended his secondary school and so on, he moved to King Edward VI School in Birmingham, which is just across the road from the Birmingham University. In fact, you can see at the back there, perceptive members of the Society can see the clock tower in the middle of Birmingham University campus. Well, he was very interested in chemistry, and he was inspired by a chemistry teacher at the school to become even more interested, and it became his favorite subject. I looked through his papers to see if I could find any record of his school days chemistry. I couldn't. I could find a page or two from his physics notebook, and I suppose reading through a few pages of it, what it does show you is he had an above-average grasp of science at that age. Well, now, chemistry was his favorite subject, and so it was only natural that when it came to go to university, he simply walked across the road to the University of Birmingham and signed up for a chemistry degree. However, when he got there, and he found he didn't like university-level chemistry very much at all. And the reason was, he says, that there was no unknowns at all. The only unknown in the types of chemistry he was doing was the yield of the synthesis at the end of the day. And what he really missed was the experimental side of science. And so when he got his BSc in 1946, he went to see his head of department, whose name was Maurice Stacey. And Maurice said to him, well, what do you want to do now, Vane? And John replied, anything but chemistry. And Maurice said, well, quite by chance, I've had a letter this very morning from somebody called J.H. Byrne in Oxford, looking for young people to be retrained in pharmacology. John writes later, without hesitation, I grasped this opportunity and immediately went to the library to find out what pharmacology was all about. And I wonder how common that is as well. It certainly applied to me during my career. So John went off to Oxford, and he spent some time with the person he later talked about as the scientist who really shaped UK pharmacology more than anyone else, and that was J.H. Byrne. This was an extremely formative part of his career. He found that experimental pharmacology satisfied his need for experimental science in a way that chemistry never had. He became very enamored of the subject. There's a funny story about his time there. Apparently, after a month in the laboratory, he was summoned in to see Byrne, and Byrne said, Vane, if you don't improve, you'll have to go, I'm afraid. John was suitably chastised, and he obviously sharpened up his act because he was still there two or 3 years later. But however, later on, comparing notes with the other people who worked in the lab, he found out that Byrne had done the same thing to all of them. And it was a sort of managerial device. Anyway, writing in Byrne's obituary, John said, those people, that's to say the people working with Byrne, each became implanted with the gene of Byrne's enthusiasm and inspiration, and they in turn passed it on to a whole new generation of scientists. So this is publication number 1 from John, and Byrne had a very simplistic way of dishing out PhD projects. He told his students to pick a drug and then to go around and test it on every single pharmacological preparation in the laboratory. And that's what he did, and John chose Paludrin, which was an anti-malarial, as you know, and tested its effect on the rabbit heart, and this work formed part of his thesis for his degree. If you read through the paper now, it has a sort of a feel of John and perhaps foreshadowed his interest in the cardiovascular system. So John graduated with another degree in 1949, and he then went off to the University of Sheffield to spend a few months with Derek Wood. This was an important year for John, but not because of Sheffield, which he didn't like at all. It was important because he met his wife, Elizabeth Daphne Page, always called Daphne, and he married Daphne, and they set up a house together, and the birth of his 2 daughters, Nicola and Miranda, followed a couple of years later. Actually, John was unhappy in Sheffield for whatever reason, and one day he got to know Geoffrey Dawes, I should have explained. Geoffrey was a physiologist in Oxford who used to demonstrate in Byrne's department. And he happened to bump into Geoffrey at a BPS meeting and asked if Geoffrey had any vacancies back in Oxford. Geoffrey Dawes, joking, said, well, I may need a bottle washer. So John said, fine, I'll take that job. And eventually the bottle washer graduated with a D.Phil in 1953. So in those days, the department was, the Nuffield Institute for Medical Research was housed in the Radcliffe Observatory, also known to the people at that time as the Tower of the Four Winds. Well, while he was at Oxford, John met an American pharmacologist called Arnold Welsh. And when Arnold went back to the US, he invited John to join him. And this led to another phase in John's career. So let's hear what Arnold said. This is a letter he wrote to me, actually, in 1997. He said, John made such a favourable impression on me, that's to say in Oxford, that I attempted to recruit him as a staff member to my new department in Yale, a position I'd been offered while working in Oxford. John, to my delight, accepted this position and became assistant professor of pharmacology and moved to New Haven in the fall of 1953. He remained at Yale for 2 years and then returned to England as a member of the Institute of Basic Medical Sciences. So Arnold was a very important person in John's life. Arnold had a different take on pharmacology to John. Arnold was more interested in biochemical pharmacology as opposed to the physiological school of pharmacology in which John had been trained. And John spent 2 very happy years there, and their friendship continued over the years and was to prove extremely important a bit later on when we come to talk about ACE inhibitors. 1955, John was back in the UK at the invitation of Bill Paton. In those days, Bill headed up the Institute of Basic Medical Sciences, IBMS, which at that time was located in Queen's Square on the top floor. And he invited John back, first of all, as I suppose a postdoc, but eventually a lecturer. And John, during this period, this was a very important time for John because he refined his investigative techniques and developed important bioassay modifications, which I'll describe in a moment. There's one study with Bill Paton I want to draw your attention to now. It was on gastric distension, and it was a study that that John came back to many years later, as we shall see, and solved an outstanding problem which this particular paper raised. So I said I'd talk a bit about John's bioassay techniques. John's bioassay technique was derived from Gadam's serial perfusion technique of 1953. This described for the first time the ability to keep tissues alive by dribbling a cascade of Ringer solution or Krebs solution over the tissue. John produced several innovations here. First of all, he put several tissues in series so that, in fact, the cascade of Krebs, whatever, was going over many tissues at once. And his second major innovation was the idea that instead of using Krebs or Ringer solution, you could use blood from an anesthetized animal. And this became known as the blood-based organ technique. And this allowed him to measure the activity of many hormones in the blood of animals, and later also of man, by the way, simultaneously and with great rapidity. And this was very important for John because he had a very impatient experimental style. He didn't like waiting for results at all. So here's a picture of the blood-based organ technique from an early sketch, and you can see it's very much as Gadam described. Ringer or blood is pumped through a water jacket to heat it up. Over the tissue, here's a mechanical lever, and over another tissue, and maybe over several more as well, before either going to waste, or in the case of the experimental animal, going back into the venous return. This became John's signature technique. It was his main engine of discovery, as we'll see, and it was instrumental in all his major research projects, including the angiotensin story, the aspirin story, and also the discovery of prostacyclin. I should say that mounting one of these experiments was a real tour de force and required a huge amount of technical virtuosity. Here's a picture taken in about 1966, which shows one of these experiments in action. You can't see, because it's down here, just out of shot, an anesthetized dog. And what you can see is the blood being piped up through this heated water jacket. And here in these plastic cups, you can see the blood dribbling over tissues. And there are mechanical levers here, and they write with a pointer on this smoke drum chymograph, which was the standard measurement technique of the day. And here you can see a blood pressure trace, which was obtained using a mercury manometer in an artery with an arterial catheter. So this was a complicated experiment to do. It involved a lot of whole animal physiology, pharmacology, a lot of patience, and they often went on for hours. If you had a good preparation, you had to stay with it. So here is a result that was obtained early with the technique, and this shows the release into the blood of a cat of histamine as measured by the guinea pig ileum after the injection intravenously of compound 4880, and you can see this contraction of the ileum here, and here's a calibrating dose of histamine. So these were John's main innovations, and these are the tools he used throughout the rest of his career to discover new things. However, back to the IBMS, more academic moves were on the cards. Paton had indicated he might go back to Oxford. John was worried about his future, and he wrote to Bill Paton to ask him what to do. And he relayed this information onto a great friend of his called Gus Bourne. John had met Gus Bourne in Oxford, and they got on really well together. And John writes, I'll read you this because you probably can't read it because John's appalling typing. But it's dated 1958, and the paragraph says, he, that's to say Bill Paton, also said that as a general rule, he liked to leave his past behind, and that if he went to Oxford, he would not automatically take this department with him. On the other hand, if there were a place for me in Oxford, he saw no reason for me not going. All slightly negative. But what it comes down to is that we get on well together, and the possibility exists that if a place is open and I'm unhappy here, he would probably invite me to come to Oxford, if only to make it possible for you and I to collaborate once more. And that's the key phrase. But as it happened, John didn't have to go to Oxford to collaborate again with Gus, because Gus took the chair after Bill vacated it in 1961, and then began a really golden age of the department, which I call the Bourne-Vane years. John, at this time, quickly ascended through the academic hierarchy, becoming a senior lecturer, a reader, and then got a chair. And between these two people, they set up an astonishingly friendly and nurturing department, which was hugely productive and turned out some absolutely cracking scientists. Interestingly enough, if you ask management theorists what they think about the idea of departments being run by two people, they rush off laughing, but I have to say it worked really well, possibly because John and Gus were like two poles of a magnet. You needed both of them, and otherwise things got unbalanced. So importantly, from our point of view, most of John's seminal work was done during this period. So 1961 came, and the department, Institute of Basic Medical Sciences, moved from Queen Square to rather more opulent surroundings of Lincoln's Inn Fields, the Royal College of Surgeons, and there on the top floor, which was vacant, the Department of Pharmacology moved in. This is a picture taken a few years later. Here's John sitting on a bench. Here's Gustav, born. You may recognize this as a young John Hughes here. John, of course, was later to go on win the Lasker Prize for his work on opioid peptides. And here, with more hair than I have now, is a very, very young Rod Flower. But this is a good opportunity to break now and show you a clip from a film that was made in 1961 for the visit of the Society to the Royal College of Surgeons for their winter meeting. I asked Gus, actually, just this morning why they made this film, and he said, well, it's very simple. We'd been there 9 months and we hadn't turned out a single paper, so we thought we'd better make something to show the Society. So the clip I'm going to show you shows John dressed up as an artist, making some corrections to a faulty chymograph tracing.
As a pharmacologist, you may work alone or with some other specialist.
There'll be some changes made today. There'll be some changes made. Going right out to buy myself a new pair of shoes. Gonna see if I can lose my blues. There'll be some changes, some rearranging. There'll be some changes made today.
So John's main interests at that time were the release and disappearance of hormones in the circulation using his blood-based organ technique. He had this particular niche of the lung as a metabolic organ, by which I mean its ability to remove hormones from the circulation as the blood passed through from the arterial to the venous, from the venous to the arterial side. He was interested, of course, in the effects of drugs on smooth muscle and also in cardiovascular pharmacology. Here's a picture of John's lab in about 1970. You'll notice that the chymograph's now given way to the Beckman multi-channel chart recorder, and instead of having mechanical levers, we had forced transducers. What you can't see is that John had mounted on this corner here at one point a closed-circuit television camera focused mainly on the chart recorder, but also on the people doing the experiment. And this enabled him, sitting in his office down the end of the corridor, to comment on the way experiments were going, to ring you up and give you advice, as he sometimes called it. And the great thing at the time was to goon around as much as you could off camera, suddenly assuming a serious demeanor as you moved into shot. It didn't always work. It's interesting these days when a PhD project application is almost as complicated as writing a fellowship application was a few years ago. And you think of all the the rules and regulations surrounding PhD studentships that we now have, 9-month reviews, yearly reviews, transfer vivas, and so on. In those days, John, I suppose, adopted Burns' technique for PhD students, and it was very simple. You went into his office, and he gave you a chart looking like this. So along the top were hormones, down the side were isolated tissues, and he said, look, there's a gap there. I want you to go away and fill it. And I know it sounds very simplistic, but it's surprising how often this led to novel discoveries and to Nature papers as well. So let's move on to John's first major discovery, and that was the ACE inhibitor story, which took place roughly between 67 and 69. So the sort of backstory here was that somebody called Skeggs had observed that there were two forms of angiotensin present in the blood, angiotensin 1 and angiotensin 2, which was a clipped metabolite of angiotensin 1, and 2 terminal amino acids had been removed. It was thought that this was catalyzed by the enzyme in plasma, which was called angiotensin-converting enzyme. However, John was interested in angiotensin, and he'd showed with Kevin Ng, who was a scientist from Singapore, and Bob Hodge, who was a visitor from Melbourne, that actually the lungs were much more efficient at converting angiotensin 1 to angiotensin 2, and in fact, that the transformation in plasma was negligible compared with the efficacy of the pulmonary circulation. Working elsewhere in the lab was this man, Sergio Ferreira. He was working on a substance called BPF. Sergio came from the lab of Rocha A. Silva in Brazil. Rocha A. Silva had discovered bradykinin in the venom of a snake, and BPF was a factor which potentiated the action of bradykinin. It stood for bradykinin potentiating factor, and it came from this snake, Bothrops jararaca, a Brazilian pit viper, and because it was probably acting as a bradykinin potentiating factor by preventing the breakdown of bradykinin, John suggested testing BPF on angiotensin converting enzyme and renin. So actually it was Mick Barclay who eventually did this in vitro in dog lung extracts in 1971, showing that this peptide blocked the conversion, and a paper with Jim Aitken came out at almost the same time showing that this also worked in vivo. So John had in his hands a tool which could prevent the conversion of angiotensin 1 to angiotensin 2, and he thought maybe this could be an antihypertensive agent. He took it to E.R. Squibb and Sons in Princeton in New Jersey because John had been consulting for them for a couple of years, and Arnold Welsh was R&D director. Arnold was very, very keen on the idea, but there was considerable resistance from other members of his team. However, they went ahead and they produced some peptide analogues. The first one was called Tepratide, you may remember it, and it did actually reduce the blood pressure in patients, but of course peptides are a nuisance to administer. Squibb had a program to look for small molecule inhibitors, but they closed it down thinking that they'd never find one, and in any case there probably wasn't a big market for it. However, and this is a lesson to all of us, there was a very diligent and stubborn young biochemist called Dave Cushman who carried on the project under the radar after they closed the project down, and he discovered captopril, and together with Miguel Ondetti he developed this, and the rest, as they say, is history. Incidentally, I'll just mention that both Cushman and Ondetti shared Lasker Prize for this discovery. So captopril was once described to me by a cardiologist colleague as the most important cardiovascular drug of the 20th century. Only a few years later, we come across what is probably John's finest piece of scientific insight, and that was the aspirin story of 1971. So again, to give you a bit of background to this, in the lab at the time was Priscilla Piper, who was a scientist who used to work with Harry Collier at May and Baker. And Priscilla was very interested in anaphylaxis and the mediators which were released during anaphylactic shock. Harry Collier was also very interested in aspirin, and he published a couple of papers showing that aspirin could block the bronchoconstriction in guinea pigs that was caused by bradykinin. And he had a couple of ideas how that could occur. They were wrong, but it doesn't matter. But anyway, the point was that this is the background. Priscilla came into the department, and she set up a perfused lung system to investigate the hormones released from perfused lungs using John's cascade technique. So this is a picture of the technique. It was a bit complicated by then. This is a photograph from a Scientific American article, but essentially the effluent from the lungs was split into two streams and went over separate banks of isolated tissues, some of which had antagonists to other hormones, such as histamine, superfused over them. I'm sorry, it's a bit difficult to see, but you can imagine with 6 tissues responding differently, it was possible to make a differential diagnosis about what was coming out of the lungs. And what they found was that in addition to the usual suspects like histamine, PGF2 alpha, they found a substance which very strongly contracted the rabbit aortic strip. This was something that they couldn't identify. They didn't know what it was. It was very unstable, and no one realized at the time that this was a product of the cyclooxygenase enzyme. But what they did notice was that aspirin could block its generation. So over a weekend, John came up with this wonderful idea, and I wrote somewhere else actually that one of the attributes of genius is to be able to see what nobody else can see, even though the facts are staring you in the face. And the fact was, everyone knew that aspirin and indomethacin could block the release of this substance. Everyone knew that there were unstable intermediates in prostaglandin synthesis. Everyone knew that prostaglandins were not stored like neurotransmitters, but they were generated as required. But it took a genius to mull over these facts and interpret them in the light of everything else that was known about prostaglandins in those days, their ability to cause inflammation and fever and so on. John spotted this connection, and he came up with the idea that aspirin blocked the cyclooxygenase enzyme which generated prostaglandins. And this was the single author paper that Humphrey mentioned. In the same year, he developed the idea further with Salvador Moncada and Sergio Ferreira, showing that it also occurred in perfused organs. And Joe Collier and I published a paper showing that it also worked in humans as well. Interestingly enough, there was another paper in Nature at exactly the same time that came from 2 other people in the department, and John had no idea until it was ready to go that they were working on it, and they showed that aspirin prevented the generation by platelets ex vivo of prostaglandins, but the interpretation on the Smith and Willis paper was that aspirin blocked phospholipase, not the cyclooxygenase. Anyway, the consequences of this fundamental discovery are still being worked through. This is the paper, and these are John's original results. Excuse me. These are John's original results, which I redrew slightly. It shows that indomethacin, aspirin, and salicylic acid all inhibit, in a dose-dependent fashion, the generation by a homogenated guinea pig lung of prostaglandin F2 alpha. This was the fundamental discovery. Interestingly enough, work from John's lab sort of anticipated the existence of COX-2, and when it was eventually characterized in the 1990s, some of the key papers on COX-2 came from John's lab. There's one up here, first author paper by Jane Mitchell in PNAS about COX-2 pharmacology, and there were others as well. Well, that sort of brings us to an end of that particular phase of John's career, and it's perhaps appropriate to hear what Byrne had to say about Vane at that time. Vane is, of course, his former pupil. Byrne wrote, "That simple methods can still break new ground and yield a rich harvest is well shown by the method of blood-bathed organs, which have lately been introduced by J.R. Vane." Well, things were changing at the College of Surgeons. Gus Bourne had taken the chair at Cambridge and left, and John was offered the post of R&D director at the Wellcome Foundation. So this is the next phase of John's career that we'll look at. So for younger members of the Society, you may not know that the Wellcome Foundation was a very important and very significant pharmaceutical company. It was started by Sir Henry Wellcome, and the interesting and almost unique thing about it was that he willed the profits from the company into a trust, and the trust dispersed these funds in the form of grants to further biomedical research. So he was a visionary man with this idea that you would have almost a philanthropic drug company. And to do this, he recruited the very best scientists he could find. Here's a picture, 1914, the Wellcome Physiological Labs. Here's Dale, another Nobel Prize winner. Barger, somebody else members of the Society may know about. And at the back, there's a very young J.H. Byrne. Actually, out of this group, you can see in front of you, 5 became FRSs. Such was the intellectual horsepower of the Wellcome, the early Wellcome Foundation. So, this is a picture of Wellcome, by the way. As I say, Wellcome had this vision of a pharmaceutical company as one where you employed the best scientists, you let them get on with things, and you utilized the results that they'd achieved to produce drugs. And this is something which he did, of course, with Henry Dale. Actually, after John's death, going through his papers, I found an interesting review by Kellaway talking about Wellcome, and it had been heavily highlighted and annotated in the margin by John, so it obviously was of great significance to him. So I thought I'd read it out to you. So Kellaway says of Wellcome, the scientific workers that he engaged were allowed to work largely upon subjects of their own choice, although he sometimes suggested topics in which he was particularly interested. They were also at liberty to publish the results of their researches. The application of this policy was in the main responsible for the scientific distinction of the workers and the high reputation of the laboratories themselves. So when John went to Wellcome, he tried to model his approach on Henry Wellcome's vision. And one of the ways he did that was by taking with him a group of people from the College of Surgeons to form a new group, which he called the Prostaglandin Research Group. And this gradually grew in size over the years and attracted an enormous number of internationally renowned scientists, and which gradually influenced the research of the entire organization. So here's a picture taken as the group got going. Here's John, Sergio Ferreira again, who was heading up the group at that time, a younger Salvador Moncada, and me with slightly less hair than I had in the first one, but more than I've got now. Elsewhere in the foundation, there were also a large number of people who members of the Society may recognize. This is standing outside the Mansion House, which is one of the crowns of that particular site. Here's Sergio again. Here's Alan Burr, many of you remember him. Over here we have Alan Green, who was head of pharmacology, Miles Wetherall and Peter Goodford, and then John, of course, in the front. One of the things John did when he got there was to try and attract as many high-caliber people as possible, both as scientists and also consultants and visiting lecturers. And this is one face that will be familiar to everyone in the Society. Jim Black was a long-term friend of John's. As a matter of fact, there's a personal memory here. I can remember setting up experiments for John and Jim when John was at the Royal College of Surgeons to measure histamine release in vivo. And John was an enormous admirer of Jim. He invited to join the foundation, which he did. As everyone probably knows, things didn't end well, and probably in a way that neither of them would have liked. But nonetheless, it was an important time for the foundation. Here's somebody more eccentric, but still lovable. This is Gladwin Buttle. So Gladwin was I mentioned him because I thought members of the society may know about him. He was Professor of Pharmacology at School of Pharmacy, and he'd been associated with the Foundation for many years as a consultant. Actually, he'd been pensioned off numerous times, but with some preternatural ability, he had some preternatural ability to know when reviews were coming up and always appeared there, whether he was being paid to do so or not. And eventually John gave up. You know, he just said, it's okay, Gladwin can come anytime he wants. And in latter years, Gladwin used to turn up with his ear trumpet, because he was a bit hard of hearing, and sit in the front row with this enormous device, a bit like Jodrell Bank radio telescope. And I remember one occasion when Gladwin asked the question to end all scientific questions. So the occasion was a presentation by a young man called Dick Hyde of compounds that shifted the oxyhemoglobin dissociation curve. And at the end of his lecture, Dick mentioned that perhaps one use of these compounds might be for trauma victims who'd lost blood. Gladwin sprung to his feet. "But have you," he shouted, "ever tried taking blood under small arms fire?" And then he went on to tell us all about his experiences in the Sudan as an army physician. Of course, John didn't just have control or responsibility for prostaglandin research. He had a responsibility company-wide. And during his stewardship, several important drugs were developed. Tracurium, developed largely by Roy Hughes. This is an ultra-short-acting muscle relaxant. Acyclovir, you all know what that is. Lamotrigine, developed by Alistair Miller in Department of Pharmacology, together with Tony Peck in clinical pharmacology, an anti-epileptic. But of course, what John really wanted was for his own group to come up with a new compound, a new drug, based upon the type of philosophy that he had about scientific research in drug companies. And this came along a little while later, 1976, and that was prostacyclin. So let's talk a bit about the prostacyclin story. So just to give you a bit of background, it had been worked out by the Karolinska group in Sweden that prostaglandin synthesis proceeded in two stages, and one was the generation of very reactive and very unstable intermediates called prostaglandin endoperoxides. More than that, they'd been able to show how you could prepare these and keep them in the deep freeze in dry acetone and subsequently use them for biological experiments. And so one thing that the group was doing at the time, this is a picture of John with Salvador, who was heading up the group at the time. One thing the team was doing was examining the metabolism of these endoperoxides in different tissues taken from different sites of the body. And what they found was that when you mix these endoperoxides with some tissues, particularly vascular tissues, the biological activity just disappeared. So here, so what was the reason for this? And here's a trace from one of the first couple of papers. And this shows you the contraction of the rapid aortic strip caused by the endoperoxides, and then it shows how this is diminished by incubation with different concentrations of pig aorta microsomes. And this is not compensated for by an appearance of other prostaglandins that could be detected in those days. In other words, a new prostaglandin was being formed. It was called prostaglandin X originally, and the original papers on this came out by Moncada, Bunting, Gruglewski, and Rod Flower, and a collaboration with Upjohn enabled us very quickly to establish the nature of this compound, which is this substance here, prostaglandin X. We now call it prostacyclin, or PGI2, and this is also short-lived and decays to this inactive compound here. And of course, the striking features of this compound were that it was an enormously potent inhibitor of platelet aggregation and a very potent vasodilator. And in a way, it was like the opposite side to thromboxane A2. And this was subsequently developed into a drug for use in pulmonary hypertension. where it is a life-saving drug. Well, here's a picture of the prostaglandin research group around about 1983. Here's John again, Salvador. I'm not in this one, but here's a young Brendan Whittle. And Stuart Bunting, who was a very talented PhD student from what used to be Chelsea, King's College, Chelsea, who did a lot of the experimental work involved in that discovery. That brings us to 1982, and this was a momentous year for John because he was awarded the Nobel Prize. Probably in retrospect, it shouldn't have been so much as a surprise, because during that summer, we'd had a sort of spy from the Nobel Assembly visiting with us. His name was Eric Angard, and he was later to become a very good friend. But he obviously came over there on a scouting trip, asking penetrating questions about research. Anyway, in November 1982, the Nobel Assembly released a press release saying that they'd awarded the Nobel Prize to Sune Bergstrom, Bengt Samuelsson, and John Vane for their discoveries concerning prostaglandins and related biologically active substances. And here's a picture of the three on the steps. Here's John on the left, of course, Sune Bergstrom in the middle, and Bengt Samuelsson on the right. It's a mark of John's enormous generosity to his colleagues that he invited several of us to go to Stockholm with him and share in the excitement of Nobel Week. And as you might expect, it was an awesome occasion. Actually, there was one amusing incident that took place. One of the duties of Nobel laureates in physiology or medicine is that they have to give lectures on their work to the Karolinska Institute. So we all trooped out there, and we listened first of all to the two talks by our Swedish colleagues, which were delivered with great dignity and in a very formal way. John got up, and immediately the mood changed. And there was one occasion he showed a slide of one of his colleagues lying on a gurney, wired up to look at prostacyclin infusion and its effect on platelet aggregation. And he happened to casually remark, as you can see, we ran out of experimental animals that week. Well, that completely brought the house down and completely changed the mood of the afternoon. And Arnold Welsh, writing later, said, John won 2 Nobel Prizes that week. Anyway, this Nobel Prize sort of, again, marks the end, really, of John's time at Wellcome, not physically, but it's sort of the end of an epoch. John stayed at the foundation for another 3 or 4 years. As you know, the foundation was merged with Glaxo in 1995, and then the name gradually vanished and disappeared from the radar, which is a great shame. During John's time, the Beckenham site was often called the University of Beckenham. But now John was almost 60, and he could have retired comfortably, but of course it wasn't in his nature to do so. And so that brings us on really to the final phase of his career. And that was the William Harvey Research Institute. So this came about in a rather curious way. On the Charterhouse Square site of Barts, which is a very beautiful site, looking more like an Oxford college than a busy London medical school, all the preclinical teaching used to take place in Charterhouse Square, but for some reason the medical school made the decision to move it all out down to another site in the East End of London. And this left absolutely acres of space. And a friend of John's, well, friend of all of ours actually, called Derek Willoughby, suggested to the dean at the time, Ian Kelsey Fry, maybe it would be a good idea to give John some space in the building since he's now out of a job. Ian did that, and John began with just one office and a personal assistant. He got some money, start-up money from GlaxoSmithKline, courtesy of David Jack, and this led off to what he calls, he named the William Harvey Research Institute. So why did he call it that? Well, let's ask John.
Have the first slide, please, which you may not be able to see very well. William Harvey discovered the circulation of blood, and he worked in many places. This is from a wall painting in Padua, where he worked for some time. He also worked in St. Bartholomew's Hospital Medical College, where I am now working. And when I started my institute, I wondered what to call it and found it didn't have a William Harvey Research Institute. And so that's why we called it the William Harvey Research Institute. In his work on the circulation of the blood, he refers to many different species, some of them shown here. But his most famous experiment was on the heart of a snake. He chose a snake because it's a cold-blooded animal and the heart beats slowly. He opened the thorax and found that when he put a clamp on the vena cava, the heart began to shrink and became pallid. And if you put a clamp on the artery, it became engorged. And what an exciting time that must have been to realize what this meant. To realize it meant that blood was going round through the heart and not ebbing and flowing. Of course, most of modern medicine is based upon his discovery. He's like an atlas supporting the whole world of cardiovascular medicine and surgery. This is the lecture theatre in Padova, where they taught anatomy using a human cadaver to demonstrate on. But in those days in Italy, it was illegal to dissect the human body. And so they used to keep a pig around so that if the police came near, they'd take the human corpse away and put the pig there instead. Nowadays, of course, it would be somewhat the other way around with the animal rightists.
So when John got there, as I said, he only had one office and a PA, but he began to recruit students. And here are some of them: Chris Timmerman, Jane Mitchell, Roger Corder, Tim Warner. They've all become professors in their own right. A couple of years later, he began to send out feelers to his old colleagues to see whether they would like to join him. David Tomlinson was professor of pharmacology at Barts in those days, so he was already there. So was Derek Willoughby. Eric Angard joined him, Ian McIntyre from the Hammersmith, Gus Bourne, his old friend from Oxford, and I became co-directors. And we obtained some funding from the Ono Pharmaceuticals, and the institute became a medical research charity in 1991. Here's a picture of us with our sponsors from Ono taken around about that time. John's main interests in those days were nitric oxide, endothelin, and COX-2 inhibitors. And in fact, I think the first international endothelin workshop was held actually at the William Harvey in 1988. I'd like to return now to that old study with Bill Paton, which I mentioned at the beginning of the lecture, to see where John had got to with that.
It's always gratifying to solve a puzzle which had been worrying you for many years. And back in 30 years or so ago, when I was working with Bill Paton, we found that if we took a guinea pig's stomach into an isolated organ bath, measured its volume, and gradually increased the pressure inside it, and here's a gradual increase in pressure in centimeter steps, there was a gradual increase in the volume of the stomach, and then all of a sudden, around about 5 or 6 or 7 centimeters of water, there was a big intake of fluid. The volume of the stomach suddenly expanded. And we showed at that time that this was not an adrenergic, it was nervous, but it wasn't an adrenergic nerve, it wasn't a cholinergic nerve. It was what became known as a non-adrenergic, non-cholinergic, or NANC nerve. And when methyl arginine came along, I said to a graduate student, maybe we can find out whether this relaxation of the stomach, which is now known as an adaptive relaxation, or if it's caused by vagal stimulation, it's known as a receptive relaxation. It's important for us to be able to take in fluid and food without increasing the pressure in our stomach. We looked to see whether this was due to nitric oxide release by a nerve. And here is an increase of pressure causing the volume increase, sudden burst of volume increase, and here is vagal stimulation causing the same thing. And when we gave methylarginine, it was abolished in both instances and came back with L-arginine supplied as our precursor. So our ability to take in food and fluid comfortably, adaptive and receptive relaxation of the stomach, depends upon nitric oxide release.
So the William Harvey today, of course, has grown enormously. We're now integrated with the Medical College. The current head is Professor Mark Caulfield. There are about 330 staff working in the Institute in cardiovascular, inflammation and endocrinology centres, and it has a strong pharmacological bias. I'd just like to say a couple of very short comments about two other facets of John's life. One was John and the BPS. John was an absolutely stalwart supporter of the BPS. He was elected a member in 1949, and he held just about every single office which the BPS had in those days. In 1984, we elected him an honorary member. He had several achievements. He introduced the published abstract in 1968, which has been a thorn in the side of all meeting secretaries ever since. He persuaded the clinical section not to part with the main body of the society in 1970, which was a great achievement, I think. He abolished a sort of class distinction between associate and full members, and he introduced the Wellcome Gold Medal Award in 1979, and fittingly perhaps the first recipient was Josh Byrne. The other thing he did for the society was he wrote incredibly witty after dinner minutes, and I'd like to have been able to read you some of these, but we haven't got time. I just want to say one other thing about John's life, and that is relates back actually to an incident which occurred in Stockholm. As we filed into the town hall in Stockholm to hear the Nobel speeches, there was a crowd of people gathered outside, sort of shivering in the Swedish winter, and they comprised well-wishers and journalists and so on, but they also comprised people with an agenda. And there was one particular group of people who shouted out to the laureates and their guests to sign a petition condemning the suppression of intellectual freedom and science around the world, including the Eastern Bloc countries. John refused to do this, and at the time it was people thought that maybe this was because he didn't care about these more global issues of intellectual freedom. But actually nothing could have been further from the truth. And I just want to tell you a bit about John's connection with Eastern Europe. So John, ever since the 1960s, had been visiting Eastern Europe as a sort of scientific ambassador. First of all, it was Bulgaria and Poland. Later, it was mainly Poland. He went every single year. He often took his wife Daphne with him. We often went with him for meetings. These were Cold War years. Poland was a very different place to what it is now. I remember, another personal recollection, I remember packing up bags containing fabricated equipment for the blood-based organ technique, the glassware I used to make and the plastic stuff, and I used to pack them all together, and together with some drugs that you couldn't get in the Eastern Bloc countries, and given to John to take over. And all this led John to be an enormously respected person in Eastern Europe, and especially in Poland. He was made an honorary member of the Polish Pharmacological Society in 1973, an honorary MD 1977, member of the Polish Academy of Sciences, and an honorary member of the Polish Academy of Medicine. Here he is with a couple of his very good Polish friends. We anglicised the names to Andrew and Richard on the left and the right, left and the middle. And these are probably two of the most important biological scientists in Poland. All this culminated in 2003 with the award by the Polish government of the Polish Order of Merit, which is a unique distinction for UK scientists. And this was awarded at a ceremony in Warsaw, attended by some of his close friends and given by the Polish Minister for Science. Again, personal recollection. All through my younger life, John had helped me with my slides, but on this occasion he was rather frail He had to give his talk sitting down, and I remember doing his slides for him, or at least helping them. So I felt, in a way, life had come full circle. So John died in November 2004, and so the last couple of slides really are his legacy to us. Well, first of all, there's the John Vane Science Centre in the William Harvey Research Institute, which he founded in 1986. All through his life, John collected pestles and mortars, and these are now on display in the lobby of the building, together with some of his medals and certificates. I should say that there's so many of them we can't get them all in. There's a stained glass window in the Heart Centre. We have a new Heart Centre on the Charterhouse Square campus, which Mark Caulfield pioneered, and there's a stained glass window commemorating his work. This is a sketch of it. And there's a bust of John made by his sculptor friend John Mills, which is in the lobby. I'd like to leave you all with something which John wrote in Tips in 1978 about his scientific philosophy, because I think it still has importance today. He said, "I've always believed in using simple methods which give results so quickly that the design of the experiment can be modified as it progresses." So I leave you with that thought. Thank you very much. And thanks to the BPS for funding this.